Welcome to this lecture on the principle of immunology. Today, we are going to explore a critical and fascinating topic, immunodeficiency. We will build our understanding from the ground up, starting with what this condition is, why it happens, and how we can mathematically measure the collapse of the immune system. By the end of this session, you will understand exactly how doctors diagnose conditions like AIDS using precise cellular counts. Let us dive in. Now that we have introduced our topic, let us start with a simple analogy to understand what immunodeficiency truly means. Imagine a highly advanced, heavily guarded fortress designed to keep out dangerous invaders. Normally, the guards are alert, the stone walls are strong, and the alarm systems work perfectly to detect any breach. In the human body, your immune system is that fortress, and the invaders are everyday bacteria, viruses, and fungi. But what if the guards suddenly fall asleep, or the walls crumble? The fortress becomes incredibly vulnerable. Even the weakest bandits can easily walk in and take over. When the immune system's defenses are broken, missing, or asleep, we call this dangerous state immunodeficiency. It lowers the body's natural shields, leaving the patient exposed to everyday microbes that a healthy person would fight off without even noticing. Building on our fortress analogy, let us look at the formal clinical definition. In clinical immunology, immunodeficiency is defined as a state in which the immune system's ability to fight infectious diseases and cancer is compromised or entirely absent. The universal hallmark of this condition is the occurrence of frequent, severe, and unusually persistent infections. These infections are often caused by opportunistic pathogens. These are microbes that live around us all the time and normally do not cause disease in healthy individuals. However, when the immune shields are lowered, these opportunists take advantage of the situation, leading to severe illness. Now that we know what immunodeficiency is, let us categorize it, starting with the first main type. Primary immunodeficiencies are congenital, meaning a person is born with them. They're caused by inherited genetic mutations that disrupt the normal development or function of key immune cells, such as B cells, T cells, or phagocytes. Because these conditions are tied to specific genes, studying primary immunodeficiencies has been incredibly valuable for scientists. By observing what happens when a specific immune gene is missing or defective, researchers can deduce the normal function of that exact gene in a healthy immune system. While primary immunodeficiencies are genetic, the second category is acquired from the environment. Secondary immunodeficiencies develop later in life due to external factors that damage an otherwise healthy immune system. The most famous example is a viral infection, like HIV, which actively destroys immune cells. However, secondary immunodeficiency can also be caused by severe malnutrition, the natural aging process, or even medical treatments. For instance, chemotherapy used to treat cancer often suppresses the immune system as a side effect, temporarily leaving the patient in an immunodeficient state. Understanding these two categories of immunodeficiency is absolutely critical in modern medicine. It explains why certain patients cannot survive common infections and helps doctors design life-saving treatments. Without this knowledge, we would not understand how viruses like HIV lead to AIDS, nor would we know how to treat it using targeted antiretroviral therapies. Furthermore, this concept plays a massive role in organ transplantation. Doctors must intentionally induce a temporary state of immunodeficiency using specific drugs so that the patient's body does not reject the new organ. Ultimately, recognizing immunodeficiency allows clinicians to intervene with targeted therapies, giving vulnerable patients a chance at a normal life. Having seen the clinical importance, we must now ask, how do doctors actually measure the severity of this condition? To diagnose and monitor immunodeficiency, especially secondary types like HIV and AIDS, doctors rely on quantitative mathematical measurements of immune cells. One of the most important metrics is the absolute CD4 positive T cell count. These helper T cells act as the generals of the immune system, coordinating the attacks of other cells like macrophages and B cells. By calculating their exact concentration in the blood, clinicians can objectively measure how badly the immune system has been damaged, removing guesswork and providing a clear numerical threshold for when a patient is in critical danger. This brings us to the mathematical formula used to calculate that exact concentration of helper T cells. The formula is C equals W times L times F. Conceptually, this formula calculates a percentage of a percentage. It takes the total number of immune cells, narrows it down to just the lymphocytes, and then narrows it down further to just the CD4 positive T cells. Let us break down the symbols. C is the absolute CD4 positive T cell count, measured in cells per microliter of blood. W stands for the total white blood cell count, representing the entire pool of immune cells. L represents the fraction of those white blood cells that are specifically lymphocytes, expressed as a decimal. Finally, F represents the fraction of those lymphocytes that specifically express the CD4 receptor, also as a decimal. If the total white blood cell count, W, decreases, or if a virus causes the CD4 fraction F to drop, the overall absolute count C will decrease significantly, indicating a worsening state of immunodeficiency. Now that we have our formula, let us apply it to a real clinical scenario. A patient suspected of having an HIV infection undergoes a blood test. We are given a total white blood cell count of 4,000 cells per microliter. Lymphocytes make up 30% of these cells, and 15% of those lymphocytes are CD4 positive T cells. We need to calculate the absolute CD4 count to see if they have crossed the clinical threshold for AIDS, which is strictly below 200 cells per microliter. First, we convert our percentages to decimals, giving us L equals 0.30 and F equals 0.15. We substitute these into our formula. Multiplying 4,000 by 0.30 tells us there are 1,200 total lymphocytes. We then multiply that by 0.15 to isolate the helper T cells. The final answer is 180 cells per microliter. Because this is below the 200 threshold, the patient has lost the critical mass of helper T cells required to mount an immune response, confirming a diagnosis of AIDS. To truly grasp the severity of the previous patient's condition, let us calculate the profile of a completely healthy university student. We are given a total white blood cell count of 7,000 cells per microliter. Their lymphocyte fraction is 40%, and their CD4 positive fraction is 45%. We want to find their absolute CD4 count. Just like before, we convert the percentages to decimals, giving us L equals 0.40 and F equals 0.45. We plug these into our formula. Multiplying 7,000 by 0.40 gives us 2,800 total lymphocytes. This step-by-step reduction mirrors how flow cytometry machines physically sort cells in a lab. Finally, multiplying by 0.45 gives us a final answer of 1,260 cells per microliter. This value falls perfectly within the normal healthy range of 500 to 1500, meaning this student has a robust, fully functioning immune system. Now that we have calculated both counts, let us compare them directly to understand the sheer scale of immune cell loss. We will set up a simple division equation to find the ratio between the healthy immune system and the severely compromised one. We take the healthy count of 1,260 and divide it by the immunodeficient patient's count of 180. The calculation yields a final ratio of exactly 7. Physically, this means the healthy student has 7 times as many CD4-positive helper T cells circulating in their blood as the AIDS patient. The healthy student has a full army of generals to coordinate an immune response, while the immunodeficient patient's command structure has been almost entirely wiped out. This stark numerical contrast perfectly illustrates the devastating biological reality of immunodeficiency. To consolidate what we have learned, let us look at this diagram summarizing both the classification and the mathematical decline we just calculated. On the left, you see a flowchart dividing immunodeficiency into primary genetic causes, like SCID, and secondary acquired causes, like HIV and malnutrition. On the right is a graph tracking the absolute CD4-positive T cell count over time during an untreated HIV infection. The vertical axis represents the CD4 count, and the horizontal axis represents time in years. Notice the solid red curve. It starts high, near 1,000, representing a healthy immune system. Over several years, the virus destroys these cells, causing the curve to drop. The critical moment occurs when the curve crosses the horizontal dashed red line at 200 cells per microliter at 200 cells per microliter. As we saw in our calculations, dropping below this threshold marks the onset of AIDS, leaving the patient highly vulnerable to opportunistic infections. We have covered a lot of ground today, so let us summarize the most important ideas. First, immunodeficiency represents a critical failure of the body's natural defense mechanisms, leaving the host vulnerable to opportunistic infections. Second, this condition can be primary, caused by inherited genetic defects, or secondary, acquired through external factors like HIV or malnutrition. Finally, we learned that clinicians do not just guess the severity of the disease. They mathematically track specific immune cell populations, like the absolute CD4-positive T cell count, to accurately measure the immune system's collapse. Understanding these principles is the foundation for diagnosing immune disorders and developing life-saving treatments. You now have a solid grasp of both the biological concepts and the concrete clinical applications of immunodeficiency. Great work today.